Recording. All right. Good morning, everyone. Welcome to Sage Live May 2024. I'm Jen Raff. Some of you might know me. I'm a senior scientist at Fermilab, which is just outside of Chicago, Illinois, and I do particle physics research, and I lead the neutrino division at the lab. So before we start, let me briefly explain what the SAGE Journey is all about and how Sage Live fits into that journey. So Sage Journey is a program whose mission is to inspire and encourage students to explore STEM careers in their future. And we do that by offering free summer camps that some of you have attended, or you will attend this summer. We also have paid internships, professional growth sessions, and many opportunities to connect with professionals who care about you and your future. So Sage Live, what we're in today, is part of the Sage Journey. It's an online gathering of students who've attended the summer camp or they're curious about the next steps in their Sage journey. And it's also a time to reconnect with Sage volunteers. We hold it monthly on Zoom between the months of August and May. So this is the last one before our summer break. And the topics that we cover are relevant to your journey. So whether it's career related, like designing your life, or a resume workshop, or how to prepare for an interview, or if it's a new and exciting topic in a STEM field like quantum computing or artificial intelligence and machine learning. So today's topic, we're going to talk about environmental science, and we have 4 special guests from Lawrence Berkeley National Lab. Each one of them is going to have about 10 minutes to present, and then we'll have a panel where we can answer all your questions. So let me start with our first speaker. Our first speaker is Colby Jardine. Colby is a research scientist in the ecology department within the Climate and Ecosystem Science Division of Lawrence Berkeley National Laboratory. And I will let him tell you more about his journey and what he does. Take it away, Colby.
Okay, great. So, so I'm a scientist in Berkeley Lab, and really what I've been doing is everything that I've been doing since I was a kid, which is being really curious about the natural world around me and using tools and friends to go and explore that natural world and try to understand how it works, particularly plants and ecosystems and how they interact with their environment. I've been for the last 20 years. Can you hear me okay?
Yeah, we can hear you. Your presentation is not in presenter mode, so if you wanted to switch to that, get a bigger view.
How do I do that? It's right here, right? There we go.
Perfect.
All right. So the area of research that I've been working on is called volatile metabolomics, or really it's the study of the smells of plants and ecosystems. Whenever you walk into, for example, a California pine forest, you smell like the pine smells, right? And if you look at the soil, you can smell the soils have different smells, and actually a lot of the organisms are releasing smells, and we're trying to understand what they are, who they are, what they're doing, and what kind of biological process they're involved in, anywhere from photosynthesis or growth or defense responses against stress. And I've done a lot of work in different field sites, particularly in Brazil, where I worked on different walk-up towers to characterize the smells within the forest and above the forest in these really diverse tropical forests that are really fascinating to work in, where you have monkeys and jaguars and all kinds of crazy animals. But just trying to understand what these volatile organic compounds are and what they're doing in the plants and the ecosystem. And the method that I like to take, where I started doing this when I was a professor in Arizona, is to work in an enclosed laboratory, say a greenhouse or some sort of controlled environment. In this case, I was in Biosphere 2 in the tropical rainforest biome, and looking at really detailed mechanistic processes that are really hard to study out in nature, and then taking what you learn from those studies at the small scale and going out to the field to try to see if you can see the same type of processes, or perhaps learn something new when you go to take the measurements in the field. And I claim to be a scienneer, which is a scientist and an engineer together. So I work a lot with instrumentation that is very specific for measuring smells in really small concentrations and how those smells change very quickly. For example, if you switch the light on, or if the temperature changes, or some environmental factor changes. And so I started building my own custom chambers for leaves and for branches and for the whole ecosystem by studying the air across those different scales. And I've been kind of doing that ever since, using different instruments and looking at different scales, all the way down from like the molecular scale, all the way up to the ecosystem scale, where we can measure these smells. So I'm just going to give you one example of a research project that was using these smells. Um, and it's a really exciting um, area of study. It's right now, it's in review in a journal, and it's a new photosynthetic pathway, which I'm calling C1 photosynthesis. So in Berkeley, California, about 30 years ago, this enzyme Rubisco, which by the way is the most, I've been reading some papers about it, it's the most abundant protein on Earth. And it's huge concentrations in leaves of plants, and also in algae and phytoplankton in the ocean. And it's this enzyme Rubisco that's taking CO2 from the atmosphere, fixing it, and creating the organic matter that we use for food and materials and so forth. And so what was discovered in Berkeley is that photosynthesis, carbon dioxide enters into the reaction, and it's a cycle. And a Nobel Prize was awarded for the discovery of that cycle. What I discovered is, flowing out of that cycle, there's like a river of carbon, which I'm calling C1 photosynthesis, which is using the same enzyme Rubisco to start the process. So I made this very simple joke, because Rubisco, it can fix carbon dioxide and it can fix oxygen. And so that's one of the reasons why there's so much of it on Earth, because it's considered a little bit inefficient. If it fixes carbon dioxide, then there's more biomass that's gained. If it fixes oxygen, then there's actually a loss of carbon during that process. And so here's just a joke. The professor is a plant asking Rubisco, okay, Rubisco, for the last time, which of these do you fix? Is it oxygen or carbon dioxide? And he's confused. He's like, oxygen? And the professor's like, no, it's carbon dioxide. So, and that is the reactions that start when carbon dioxide are assimilated by the plants. And one of the VOCs, everybody's heard of ethanol or alcohol, but one of the most simple alcohols with just one carbon atom is methanol. And plants are releasing the methanol back into the atmosphere as they grow and as they assimilate the carbon dioxide. And I've been studying this methanol being released from plants as they grow for the last 20 years in Europe, in the United States, and Brazil, and just wondering to myself, like, where is all that carbon coming from of the methanol, and why are the plants releasing so much of it back into the atmosphere? And so I just was thinking about that for so long, and I did some studies to show that the methanol is coming from the cell walls, and it's made from this really exciting process, which I'm going to show you in animation, so I better stop talking in a few minutes here, of this photosynthetic C1 pathway, where you can see in the chloroplast, the carbon dioxide entering into the Calvin cycle, and then flowing out of that Calvin cycle you see these amino acids like serine and methionine. Methionine is an amino acid which is well known for making proteins. But I've found that the head of the methionine, you can see that blue head on the methionine, that came directly from carbon dioxide seconds before. And so then the methionine, it gets activated by reacting with ATP, and then it can donate the methyl group, that head, to thousands of different things during the growth of the plant. One of the things that it's donating that head to are the growing cell walls. So like the pectin that get packaged up in these vesicles and sent out to the cell walls, they get loaded with the new with these heads of that same carbon methyl groups. And when the cell wall decides to grow, it's going to release that blue ball, that blue methyl group, as methanol. And then the methanol will go back into the atmosphere as reporting that the cell is starting to grow. And so so this is the overview of the photosynthetic C1 pathway. It's it's called C1 photosynthesis because it's just involved a single carbon atom that comes from CO2. And everybody, or a lot of people, have heard of C3 photosynthesis, and that's what most plants are doing. Why is it called C3? Well, you can see within the Calvin cycle, these intermediates with 3 carbon atoms, like the 3-PGA, the 3-GP. And so that's how photosynthesis is recognized now, C3 photosynthesis. So now there's this new process, C1 photosynthesis, which is happening together with the Calvin cycle. But I found that it's actually not new. It appears to be present in all plants, even in cyanobacteria. And perhaps it helped the evolution of photosynthesis 2 billion years ago when cyanobacteria first started to create oxygen from the water splitting. And so let me just stop talking there and just show you an animation of this this new photosynthetic pathway, new to science, but it's actually a very ancient process that perhaps helped shape the evolution of life on Earth. So it starts with carbon dioxide going from the atmosphere into the land. So it's going into the oceans, but it's also going into the land. You can see the carbon dioxide finds its way to a leaf in a forest, goes into a stomata, and then it goes into a photosynthetic cell. And again, it's going to enter into the Calvin cycle here. It goes into the Calvin cycle, which requires light to power that fixation of CO2. And then it's going to go through a series of enzymatic reactions, starting with Rubisco. And there's 7 different enzymes that are going to take that carbon atom and transfer it. I see this kind of like a game of soccer where you just have a ball of soccer and you're just passing the soccer ball from one player to the next. Gets the soccer ball. Okay, now I got it. Now I'm going to pass it to you. You got it now. And in this analogy, scoring a goal is the final guy who gets the soccer ball. Methionine, you can see here, methionine has the soccer ball now. He's going to score a goal by passing that methyl group to something to help grow. In this case, it's going to be the pectin on the cell wall. So it gets passed to the pectin on the cell wall, and then that gets transported to the external part of the cell and the cell wall. And when that cell wall starts to grow, some of those blue balls are going to come off as methanol. And that's really important for controlling and regulating the growth process. And so now we can see photosynthesis on Earth happening only in the sunlit section of Earth, whereas the methanol during the growth can happen in the sunlit portion or also at night as the plants are growing and releasing the methanol. But photosynthesis can only occur during the day. Thank you very much.
Thanks, Colby. That was very interesting. If we have maybe one question, we could take that now, or you can save it for the end. I'll check the chat. Okay, we'll take questions at the end. Thanks again, Colby. All right, so our next speaker is Lauren Louis, and she is a research scientist in the Environmental Genomics and Systems Biology Division at Lawrence Berkeley National Laboratory. She uses environmental sequencing and computational biology methods to study the impact of microbial species on global element cycling and their feedback response to climate change. Currently, she studies sites in the San Francisco estuary and the Baltic Sea. That's cool. And Lauren gets excited about how we can understand the response of microbes to environmental change just from the information contained in their genomes. So, Lauren, welcome. Take it away.
Thanks, Jen. Let's see, let me just get my presentation going. Okay. Can you see my presentation?
Yes, looks good.
Okay, great. Okay, so I'm excited to talk to you about my research today and a little bit about my journey as a scientist. So this was a tweet that LBNL did on me. And I think it just really illustrates kind of where I come from and how I got interested in science. When I was little, my grandpa wanted me to learn how to type. And so he taught me how to use MS-DOS. And so to actually start the program. And that was kind of the first time I was like, oh, I can tell the computer what to do and it'll do what I want it to do. And then when my mom also taught me a lot about chemistry and biology when I was cooking and gardening. And so I was interested in both. And I actually didn't know what I wanted to major in. So I started out as a math major and then that led into using computers to study biological systems. And so that's kind of what I'm going to talk about today. Um, so kind of the big thing in my research right now is I generated a lot of DNA sequencing data. So there was one day where I loaded all of these, so all these little squares are what they call flow cells, where you can load DNA and get the sequence out. And so if you're not quite sure what a terabyte is, if you have a laptop generally has 50 gigabytes of storage space now. So that's like about 100 laptops worth of data. And so I'm going to talk to you about how I got to this point where I sequenced so much in one day. And so my study area is the San Francisco estuary. And so I grew up in Sacramento, and so I've been really excited to study what's happening in the water here. So if you're not from the Bay Area, the San Francisco estuary is composed of the Sacramento-San Joaquin River Delta. So those are the two main rivers: Sassoon Bay, San Pablo Bay, and San Francisco Bay. And so as you can imagine, with millions of people living in this area, the water resources are very highly impacted by all the human activities. So all of the wastewater treatment, agricultural runoff, recreation, that all flows into the San Francisco Bay and the Delta. And so it behooves us to take care of this natural resource because we do use the water for agriculture. It actually supplies 2/3 of the drinking water to the state. So Southern California actually takes a lot of the water from Northern California. And, we should just be environmental stewards. And so that's why I'm studying the estuary. Another thing about the estuary is they don't understand why it's not experiencing what they call eutrophication. And so an example of eutrophication is in the top right corner. Basically what happens is eutrophication is when a lot of the algae start to grow. And then that takes all the oxygen out of the water. And so that kills fish, kills the other organisms, and then it creates a bad feedback loop, and it just basically destroys the balance of the ecosystem. And so they know that things like nitrate and phosphates can cause this because it's just a lot of nutrients. And so nitrates and phosphates are very common in fertilizers. So agricultural runoff can be a big contribution to this. But the San Francisco estuary isn't experiencing as much as other places. And so they think it's because it's actually a very turbid estuary. And so when you get runoff from the rivers, there's a lot of silt, and that prevents the sunlight from penetrating and causing the algae to grow. But as we've been having more drought, that we've been having more of these harmful algal blooms. And so that's happened in Lake Merritt and Oakland in 2022, and also a little bit in 2023. And so these are happening more often. And so I'm trying to help figure out, is there a way that we can help study what's going on? One way that the city of Oakland has been trying to mitigate this is by putting in fountains to actually oxygenate the water. But this one year when it happened, thousands of fish died. It was really bad. And then this is just a microscope picture I took of. I actually was able to get a sample. It's it actually has a little flagellum and spins around, so kind of cool to see. But very damaging to this the system. So I'm really interested in the microbiome because I was talking about the nitrates and the phosphates. So the microbes take part of the nitrogen cycle that only they can. Many of these places in this nitrogen cycle diagram that I'm showing you where it's anoxic, so the bottom half, microbes take care of that. So, Colby talked about the carbon cycle, nitrogen cycle is also really important. And so one reason also we're so interested in the nitrogen cycle is nitrous oxide is a greenhouse gas. And so if the microbes can't finish the cycle, then we get more and more nitrous oxide released into the atmosphere. And so we want to try to study what they're doing and when they do it. So the cycle keeps on going. So the issue with this is that because they're so tiny, right? And we actually don't know how to grow them in the lab. We don't know what they're doing and we can't track them. And so that's why I do environmental sequencing. So if you just get a sample and sequence the DNA, you can actually see what's in there. Just from the DNA, you can figure out the taxonomy. You can kind of count them. You can look at their genes after you sequence their genomes and assemble them. And so it kind of looks like this for this project. You collect the water, filter the water. This is what a filter looks like. You take the filter off, you try to extract the DNA from that, and then you do genome assembly. So that makes it sound really easy, but this is the main challenge right now in this part of the field. Because we have these genomes, we can see what their genes are. So Colby's animation was really great because all of those genes encode those proteins, so we can kind of guess what they're doing and where they might live. But we often don't have complete genomes because of the sequencing technology. And so I like to use this puzzle analogy for environmental sequencing. So basically imagine you have all of these different organisms and their genomes. Each puzzle is a genome. You dump them in a box, and then you try to put all the puzzles back together. So that's hard in itself. The environmental sequencing, because of the way the technology works, is actually like putting all those pieces in a blender and shaking it up and then trying to assemble the genomes. And so you can imagine it's very difficult. And so that's why I use the computer to help assemble these genomes. And so the dominant technology, which I'm just going to call short read technology, has given us a lot of insight into environmental microbiomes. But now we're switching over to this long read technology, which is what I'm focusing on. And so just to visually see the difference, the one on this is an assembly graph. So this is after you put all the pieces of DNA together and visualize it. The left side is the short read sequencing technology, and the right side is using long read sequencing on the exact same sample. And so the microbial genomes are often circular. And so I've highlighted them here. You can see that they're popping out as circular. So that's basically what we want. So I'm collaborating with USGS. They do monthly sampling cruises and they're giving us samples. And so that's how we got to here. And so we are pulling out genomes of really important marine microbes that haven't had their genomes finished before. And I'm in the process of analyzing this and seeing what they do and where they are located in relation to environmental data. And so for a comparison, I also have this collaboration with a group in Sweden to study the Baltic Sea, which is another highly impacted estuary. So this comparison can help us figure out more and understand more about how microbes are impacted by human activities. And so I just want to thank everybody on this project that helped give me samples and worked on it with me. And I'm happy to take any questions.
Excellent. Thank you, Lauren. Do we have any questions from, oh, no, I saw Colby's clapping hands and I thought that was a question. Are speakers allowed to ask speakers other speakers questions? I think so. All right, I don't see any questions right now, so we can move on. Thank you for the.
Could I ask a quick question then?
Sure, yeah.
Yeah, I was wondering about with the sequencing, like with the long read sequencing and then the short read sequencing, which one is better? Is one of them like way more expensive than the other one, or do you use both?
Yeah, so I actually use both. So the short read sequencing, it's cheaper and it's easier because you don't have to be as careful with the DNA. If you're doing long read sequencing, the DNA extraction is a lot more difficult because if you don't have long pieces of DNA, you can't get long pieces of DNA sequence out from the sequencing, and so that's been a challenge, and that's also something I've been working on. And then, but in general, the technology has gotten good enough where the genome assemblies are make it a lot easier. The error rate of the sequencing used to be a lot higher for long read sequencing, and so that was actually made it harder to do the assemblies. But now, because it's been improving, it's actually it's better. But yeah, if you're limited by cost, then then people still do the short read sequencing. So
Is the short read one also faster? I mean, it seems like it would be if it's a shorter piece of DNA.
actually the long read sequencing is in real time.
Ah.
And so some people are actually trying to use it in the field now. So the nanopore sequencing, they've been developing a device where you could just hook it up to your computer and then put the DNA in. Like, so if you're working in the field, you could do a really quick DNA extraction and then put it in the device and try to see what it is. So this would be really useful for people trying to track pathogens. And so that's actually something that people are actively working on. Yeah.
Very interesting. All right, I'll ask once more. Any questions from the audience? All right, then thank you again, Lauren, and we'll move on to our next speaker. So next we have Dawn Shinicki, and she's a research science in the Environmental Genomics and Systems Biology Division at Lawrence Berkeley National Laboratory again. She's a Bay Area native and received her BA from UC Berkeley and her PhD from UC Davis. She works on finding bacteria from agricultural fields that eat climate-changing gases. And she uses a combination of computation and engineering to study the bacterial genes. Currently, she's focused on rice fields in the Delta and in Northern California, as well as sorghum fields in Central California. Dawn loves the part of her job where she gets to see the diversity of bacteria that come from each environment and see what special functions they have in the environment and can have in agriculture. So welcome, Dawn. Thank you. And whenever you're ready, you can go ahead and share.
Okay, thank you for having me. Is this good? Can everyone see?
Good. Looks good.
Okay, I really enjoyed hearing everybody else's talks too. So I'm going to be talking a little bit. I thought I'd talk first about how I got into what I do now, and where I came from in the Bay Area. So I grew up in the East Bay, and I graduated from UC Berkeley back in 2004, and that at the time I thought I wanted to be pre-med because I just had this sense that I wanted to like help the world somehow, and I hadn't really met a scientist before. So but then I did a little bit of lab work, and I just changed the whole career path that I had. Just fell in love with research and trying to figure out, pushing this line of what we know and what we don't know across the world to a little bit more that we know. So I started out I went from my bachelor's to my PhD, where I looked at how the sugars in plants can make a plant a better biofuel crop or not. So that was, and that was in rice. That was great. And then I moved on to do a postdoc where I looked at how these sugars can make something more or less resistant to plant disease, which is a big issue in agriculture too. So, and I looked at this powdery mildew pathogen that attacked the plants, and you see that in like grapevine and vineyards up in, and other plants as well. I then went on to my next job, where I started to look more into microbiome science and studying how the sugars that are released into the soil can create different microbiomes. And it's kind of like what you eat into your stomach microbiome can be different. And it's true for plants too. They have their own microbiome around their roots. So from there, I did do a little bit of a stint where I tried to start my own company for a little bit. And before I decided I didn't want to do that, but this was a picture of me pitching my idea to Y Combinator, where it's like a venture capitalist area where all these wealthy donors join to see the next big idea. And at the time, I was really excited about studying microbes that could replace fertilizers, since fertilizer runoff is a big issue for groundwater contamination. And that was really exciting. And I think I learned from that I didn't want to just run my own company. I just wanted a regular job where I showed up every day. So that's where I ended up where I am now at Berkeley Lab. And I still think about the company idea in the back of my mind, and I may still come back to it. But it has influenced what I study now, which is looking at little bacteria that live in the microbiome of these plants. And many of them have important roles in the environment. So lots of them eat these gases like methane. Methane is like even more strong than CO2 in how it warms the planet. So it's 40 times more strong than carbon dioxide. So it's really strong in its warming potential, and it has a very strong impact like in the near term, which is what we need to focus on because of climate change being so, so urgent in addressing in the environment. So I study these microbes that can eat it and maybe engineer them to be better at it. So I also look at microbes that live in the environment that eat nitrous oxide. nitrous oxide is even more potent than methane in its warming potential. So it's 300 times more potent than CO2. And it's very, when you look at agriculture, like replacing biofuel crops, it's definitely more sustainable, but there's this negative impact from agriculture on the environment too that we have to consider. So that's what I'm trying to do with my research now. And this is kind of just an overview. I'm not going to go into too much detail, but one of the things I do is I find these microbes, I engineer them. So you can engineer microbes by different ways, but we do something called bar-coded engineering, where you're basically putting in a code and you're engineering it into a microbe. And we do that with microbes that we can isolate, native ones. And that goes into their genome, and we can then track it computationally. And we can do machine learning to look at, oh, what does this gene do? What does this gene do? And kind of see what pops up as important. So that's kind of what I'm working on now. And just thought I'd talk a little bit about what it's a day in the life of a scientist. We all kind of work in this really nice building. Go for a run in the morning, come into the lab, and we work on your own a lot of the time, but sometimes you work with other people. But often I just have my headphones on, I'm listening to like music or like a book or something, and then just like getting my work done, and then you finish your projects and you go home. So that's kind of what it's like in the lab. It's very chill, very fun place to work. I just like the work a lot too. So, and that's all. Thank you.
All right. Thank you so much, Dawn. Do we have any questions either from the audience or from the speakers to Dawn? Colby.
Hey, Don, that was really cool. I was just wondering about the diversity of the microbes there in the soil. I saw that I think it was Pseudomonas or Streptococcus that I saw there, because I know that's like an infective bacteria in people.
Yeah.
Is there
Yeah.
like, I guess my question is about like the diversity of the soil microbes, and like, do they have a similar microbiome that humans do, or is there very different bacteria in people versus like in the rhizosphere?
Yeah, that's a great question. They're different bacteria. So if you were to sequence them, what's in your stomach, like you can just take a sample and sequence it kind of in the way Lauren described. You get this big blender of DNA that you can assemble computationally, but you would see that it's different than what you would see on the plant root. But it depends on how deep you sequence. So if you just look at like a species, like a Pseudomonas is one level of knowledge about an organism, and then like we are Homo sapiens or whatever. So if you're just looking at the first part, then yeah, there's like Pseudomonas aeruginosa, which is a pathogen in humans. But the one we were looking at is closer to Pseudomonas fluorescens, but we think it was actually helping the plants, and it was being recruited by the plant with its sugars. So it was, saying, here, come and eat these sugars and I will use you somehow. So that's what we think was going on there. Although that's still just in the works. There's still more articles coming out about about this particular Pseudomonas and what it was doing in the plant root. It was a recent discovery that this happens in like corn and also in sorghum. So yeah.
Really cool. Thank you.
Yeah, very cool. All right.
I see a question in the chat from Loy. What was it that made you switch away from pre-med? Yeah, it's funny. Just working in the lab, I really enjoyed it. And I remember we had a professor who he was retiring at Berkeley as my undergrad, and he wanted to still come into work and do research, even though they weren't paying him. And he wanted to, move into this like basement of the greenhouse, which was the creepiest place that I could think of. Is like all these creaky noises, no lights, and we were helping him set up his lab bench. And I just felt this like, wow, like this is what somebody does who truly loves what they do, that they're just, and I worked in the medical field for many years. I was an EMT on an ambulance. That's how I paid my way through my undergraduate. And I never saw anybody that dedicated. A lot of people are very burnout and very tired, and they're ready to go home. They're not going to come in for free. So yeah.
Are you scoping out spots that you will take up residence when you decide to retire? The creepy basement of
Yeah, I don't know that Berkeley Lab is as generous with like the people coming in. Who knows? I don't know. But at UC Berkeley, they did give retirees like a little office. They don't guarantee where it will be, but yeah. I wonder about that. But I think when I retire, I'm retiring. But I have a plan to be like, maybe be a yoga instructor, in my next career or something. I'm just going to spend a lot more time outside and, with people. So, yeah.
Excellent. Thank you so much. That was fantastic. All right, so let us move on to our last but not least speaker. This is Andrila Mukhopadhyay, and she is also a senior scientist in the Biological Systems and Engineering Division at Berkeley Lab. She's the principal investigator of her team that's part of several large interdisciplinary projects, mainly focused on engineering and environmental microbial systems. She's the vice president of the Biofuels and Bioproducts Division at the Department of Energy-funded Joint Bioenergy Institute, and her group uses a range of functional genomics, metabolic modeling, and systems biology approaches. Her group specifically focuses on developing robust strains that show high tolerance and productivity during biofuel and chemical production, and the optimization required to achieve scalability. So let's hear from you, Andrila.
All right, I'm going to share my screen. Looks good?
Yep, looks great.
All right, so a lot of the work that was described so far feed into what I'm going to tell you about today, but it's a little bit different. So I wanted to start with sort of my realization when I first came to Berkeley Lab, where that symbol actually came from, and that it depicts the light source and the clock tower down the hill, and that it's not a made-up image, it's a real thing. So I always like to start there because I thought that artwork was super cool. And so I went to high school a really long time ago, but I did do some experiments when I was in high school. This was so many years ago. And I remember all the time, all the grown-ups around me would talk about either having tea or coffee. And all the time they would say that, oh, I don't think tea is going to cut it. I need some coffee. I need to stay up to do this task. And I wondered, like, based on what are they saying this? They can't drink the tea or coffee both at the same time to figure it out, because then they would have drunk both. The order doesn't matter. So my high school experiment was to try and figure out how much caffeine there really was in a standard cup of black tea versus instant coffee, which is what most people around me drank. And I figured out by begging and insisting at a neighboring lab that the equipment you need to use to extract caffeine from either tea or coffee would be the Soxhlet apparatus, where you put your material over here and it kind of sends down solvent over and over and over again, your material, and finally gives you a distillate with just your extract, and you can dry it down and you can weigh it. I got terribly variable results every time I did this work. But in general, there wasn't that much difference. And so I felt like all of these people that they were talking about drinking coffee versus tea didn't really know what they were talking about. So that was my first experience with a science experiment. And this I did in a small school in Pune, very close to Mumbai in India, back in the late 80s. And Pune is a city that is a smaller city in India, but it would be considered pretty big by American standards based on the population. So then that kind of led me to my rest of my journey. I went to college in a place called Lucknow in India. It's a very historic place. After that, I came to IIT Mumbai to do my master's. From there, I made my way to Chicago to work in the lab of Dr. Lin, where I got introduced to a lot of cool bacterial systems. When Professor Lin moved to Emory University in Georgia, I moved with him. And from there, I came to Lawrence Berkeley National Lab and UC Berkeley to work in Dr. Kieslim's group. And I basically never left. And people talked about retiring. I think I'm going to stay the rest of my life here from the way it's going. I've been here many years now. And so this has sort of been my path from that first high school experiment to today. And I wanted to share with you my favorite quote when it comes to why people become scientists. I'm going to read this out because this is a good one. Scientists are people with very dissimilar temperaments doing different things in very different ways. Among scientists are collectors, classifiers, compulsive tidier-uppers. Many are detectives by temperament, and many are explorers. Some are artists and other artisans. There are poet-scientists, philosopher-scientists, and even a few mystics. Obligative scientists must be very rare, and most people who are in fact scientists could easily have been something else. So this is something that's very inspiring to me because I think I'm one of those people who could have easily been something else, and yet I am doing what I'm doing, and I really enjoy it. And so the reason I came to Berkeley Lab is because when I came here, I discovered this whole idea of team science to achieve big goals. And this is a quote by Ernest O. Lawrence, who founded this lab. And the heart of this quote is that science isn't done alone. Science is done by lots of people achieving lots of small successes, and we stand on top of those successes. And the smoother your science is going, the more people have contributed to it, and that's why it's going smoothly. And so this was very inspiring to me because I felt I could make a small contribution and it could be meaningful. So this is how I landed up at Berkeley Lab and being part of these very, very big projects. I'm part of many big projects with all of the folks you heard from before. And today I'm going to tell you some of the work we've done as part of the Joint Bioenergy Institute. So this is an institute that focuses on developing the scientific knowledge and figuring out what new technologies are needed, what are the gaps, how do we fill them to convert the maximum amount of carbon we can get from a renewable carbon source into biofuels and bioproducts. So why are we doing this? I'm going to show you some graphs. The takeaway from these graphs is that the big factors that are contributing to our greenhouse gas emissions are transportation, and energy generations. These are the big hogs of the contributors. And in that, energy and industry has a very big role to play. So if we want to take a big swing at this problem, this is a place where we could have a huge impact. So what does that mean? That means that what we are currently getting our energy from, a barrel of oil, that needs to be changed into something else, if not fully, at least partially, as much as we can. So in the US, petroleum is the primary source of transportation fuels and chemicals. So not just the gasoline that you put in your car, but also diesel fuels and jet fuels and everything else. So if you're wearing shoes right now, or glasses, or using a computer, or using a pencil, or notebooks, or a handbag, Anything, look around you, 90% of it has some component made out of petrochemicals. So the goal here is we want to supplant as many of these compounds, biofuels and bioproducts with biologically made compounds. So a bunch of structures for you to look at, just to give you a sense of how many different compounds there are that we could potentially make. Right, and I'll come back to this a little bit, so you'll see these structures a little bit more. So one thing I wanted to tell you is sustainable aviation fuels, which were these sort of like cyclic compounds up top. These are kerosene-like compounds, polycyclic compounds that have so much energy in them that when they split apart, they release a lot of energy. These energy-dense compounds are sustainable aviation fuels because you can get them from renewable carbon sources. And they are sort of like a key piece in our ability to lower greenhouse gas emissions. So, this red line shows you what happens if you don't change anything. And then in order to be able to make a big impact, you need to incorporate sustainable aviation fuels in your configuration of life moving forward. Not only that, you can't really supplant petrochemicals unless you replace a heck of a lot of other things that come out of petrochemicals as well, such as plastics, paints, building materials, polymers, pigments, resins, coatings, all of that. And so the goal here is to use microbial strains to make these kinds of compounds. So the question is, can we use microbial platforms to make the kind of compounds we get out of a barrel of crude oil? And the answer is yes, we can make these kinds of sustainable aviation fuels. And these sustainable aviation fuels, because this is a new technology, you have an opportunity to make something that's even better than what you have today. So here is an example of Jet a fuel that is used currently in aviation, and an example of what you get mathematically if you calculate the distance you would get if you used a sustainable aviation fuel that was designed to be highly dense. Not only would it improve the economics of the process, it would improve range, which means for the same amount of fuel, you can go a larger distance. You can see that these small differences, especially in a large industry like aviation, they can have tremendous impact on efficiency and lowering our greenhouse gas emissions. So this is well worth doing. And when you go out there and tell people this is something exciting, they'll be like, who even does this? Are there any examples? This is just, whatever, pipe dreams. Nobody can ever pull this off. So I wanted to give you a few examples. There are companies right now doing this work. One is called Lancetech that does microbial conversion of sustainable carbon sources to ethanol and then converts that ethanol to jet fuel. And then there's Genomatica that's making a polymer precursor called BDO using microbial platforms. These are just two of many examples, but these are my two favorite examples because they have scaled up and shown success, and I wanted to give those to you. So here at Berkeley Lab, we are trying to use these microbes with these versatile metabolisms. One of them is the Pseudomonas that came up earlier. It's not the fluorescence that is found in the soil where Dawn is looking, and it's not Aeruginosa, which is an obligate human pathogen, but rather it's a completely other strain that has its own metabolism and its own profile. The other is an oleaginous yeast, which means that it has a natural propensity to accumulate oily material and is a good host for these kinds of purposes. So what we do is we take these organisms and we study their genomes and their metabolisms, and we manipulate that metabolism to make it eat the carbon sources we can give it from a sustainable carbon source and make it produce these products that we really want to supplant petrochemicals with. And this can be done. So I'll give you one example, and this is an example of a compound that looks somewhat like this. This compound is very, so before I get into that, I wanted to tell you a little bit more about this compound. Yes, this compound is very similar to this molecule, indigo. Indigo is a molecule used to dye your jeans. I'm sure several of you are wearing your jeans right now. Let me go back to this slide. So in this slide, you have a breakdown of what it takes to make a pair of genes. It gives you both the numbers for what it costs to produce that pair of genes, the fabric, the harvesting, the transportation, the water use, the chemicals, the dyeing process, the waste disposal, all of that, and the amount of carbon emissions and water you will use in the lifetime of that gene. It's a pretty substantial amount. I can share these infographics with you later, but suffices to say that these kind of molecules are very energy intensive to make, and they create a lot of polluting streams that are hard to manage. So being able to make these compounds biologically and safely and coming up with different processes for around these kinds of industries can be very impactful. So here is a picture of some organisms that we have engineered to make this compound. You can see that they're making so much of these compounds that they've almost turned blue-black in color. And that's pretty cool. So we took our Pseudomonas organism and we did these genomic modifications and the metabolic rewiring that I told you about. And we were able to show that this organism can now take a sustainable carbon source like an aromatic molecule or a sugar and make a lot of this blue material quite high. These are all open source papers. So if you're interested, you can get in there and read the nitty-gritty of it. Next, we wanted to show that we can actually scale up this process a bit. We have access to some nice process development units at Berkeley Lab. So with the help of scientists in that group, we were able to show that we can make larger sized culture that still produce this blue molecule. And at the end of the day, we were able to get a lot of this is all live bacteria producing that molecule that you can then extract and separate. And now you have this powder version that you can use in your downstream applications. So coming back to be able to use your microbial strains for making a large number of compounds, I think we can do a lot of good over here. And I was thinking that if any of you are interested in learning more about this, we do have a program in our lab that takes in high school students to teach them various parts of this process. It's a very small program, but it's very intensive, and there is a very specific criteria for the incoming students. But if you're interested, do check it out at iclem.org. So ICLEM stands for Introductory College Level Experience in Microbiology. And that's what I had for you today. I wanted to show you the big, large group that we are part of, and also my research group where we work together to engineer these organisms in the lab to make these kinds of useful molecules that can replace petrochemicals. And with that, I will take any questions.
Thank you so much, Andrea. Are there any questions from the audience or the speakers? Oh, I have a quick one. You mentioned this company, it was called LanzaTech, and the other one that are making
fuels and polymer precursors, yes.
Are there any airlines that are actually using that yet?
Yes, so the LanzaJet fuel definitely gets used. And I don't want to give an off-the-cuff answer that might not be correct, but this is commercialized. There are other airlines that have blended in bio-based fuels into their mix. So one very interesting thing about fuels is that it's rarely a single compound. Here is the reason for this. So imagine your family was starting on a road trip from Alaska, and you're driving down the continent all along that beautiful highway all the way down to San Diego. Think about the altitude, the temperature, and the humidity that's going to change all the way along the path. The same compound cannot maintain its properties as a fuel to run an engine that transect. But have you ever had to think about it? No, you just stop at a gas station, fill it up, go. Fill it up, go. This is because fuels are a blend, and they're blended to maintain that property. And of course, the components of the blend are very, very strongly kept trade secrets that we will never learn about, but they're blend none the same. But the nice thing about them being a blend is that if we can make bio-based components that can serve as blends, we can begin to to replace many components of that fuel. In fact, we can pick and choose components that improve the fuel performance, reduce carbon emissions and soot formations, and reduce the use of chemicals that are particularly environmentally impactful to produce. So we have a couple of different needles we can move on that front.
Yeah, that's fascinating. Thanks. Okay, I see Sydney wrote a question in the chat, which is, she said, you lightly touched on clothing impacts such as jeans. How do you feel, or any of the guest speakers, about fast fashion companies? Good question.
That's an excellent question. The market is responding to our demands. As people become more educated about the impact of what it is they're doing, the market will also shift. The market will shift for two reasons. There is something better, cheaper, and more appropriate for your needs out there. And you care, and you're willing to make adjustments in your lifestyle to accommodate that. So it's a two-way thing. So there is a very famous saying that was used by an economist, Thomas Friedman, but the saying actually comes from a sheikh in the Middle East, which goes something like this: that human beings did not stop using stones because we ran out of stones. We moved into the industrial age because something better and more efficient came along. So as scientists, it is our job to provide these more efficient solutions. And then as a consumer, it is our job to move away from these fast fashion and use the good product. So that's my answer, that yes, the quick answer is fast fashion is bad for the environment. But the more complicated answer is anything better is going to be so much more expensive. People can't afford it. What do you tell those folks? They need what they need as well. And culturally, we need to shift towards that.
Thank you. I see also Loy put a question. Are the traditional petroleum companies vested in your research?
Several are. They do see the writing on the wall, and they see that this is worth investing in. They want to have a diverse portfolio and a stable investment into products. And some of them are genuinely motivated in doing the right thing, or at least they've created portions in their company that are. So as a great example, just in the previous talk, Dawn showed you a picture of her and her colleagues sitting at an institute called EBI. EBI was funded by British Petroleum. British Petroleum had some financial problems later on and could not continue that funding. But as my career in this field progresses, some of these work goes hand in hand with them. I'll give you another example. Say tomorrow I produce a molecule that in the lab has all the properties of being the next best biofuel blend. I need to go through a tremendous amount of regulatory check-ins before anybody will agree to scale it up and produce. Well, who will do these check-ins for me? It is actually these petrochemical companies that will step in. Aviation companies will step in, testing facilities will step in, and they will do the research. A lot of them will start becoming government mandated to do this work. Not just petrochemical companies, but other federal entities like the Department of Defense is very interested in these kinds of research as well. So yes, there is a lot of interest in the work that we do. It's slow, but it's there.
I'm very glad to hear that, because I always feel like the traditional companies are just going ahead destroying the planet without any any feelings in either direction. So I'm glad to hear
those are big ships, right? They're hard to turn around. The public in general wants one thing, but then behaves in a different way, right? Next time we are wandering through our house, it's a really good exercise to see how many products we use palm oil. And then you will get a sense of how much an individual consumption of palm oil is. Why am I bringing palm oil? It's because entire forests have been raised to the ground to grow palm trees to meet our palm oil needs. These are monocultures that are so susceptible to all kinds of invasive species, and they've destroyed entire ecosystems to make it. But nobody made this just by themselves for fun. They made it for us. We are the ones using that product. So it's a give and take thing, and it moves slowly, right? The technology has to be there, the need has to be there, the education has to be there, and then all of this has to kind of come together in cycles to move forward.
Fantastic. Thank you so much. All right, we have a bunch of questions that the participants submitted when you registered. So let me start asking those as well. So the first question I think we will direct to Lauren because it seems directly relevant to her. So the question was, what areas of environmental science most commonly have people with math degrees? And I remember Lauren said that she does have a math degree.
Yeah, so the most common places, if you have a math degree for environmental science, are ecology and bioinformatics. And so that was what my PhD was in. So for ecology, there's actually quite a bit of math and modeling that goes into understanding how different species interact, whether this be microbes or macro organisms, as I would call it, things like endangered species, tracking, epidemiology. All of those actually use a lot of math and modeling because it depends on the probability of organisms interacting with each other. And so that's where the math comes in for that. For anything with looking at the genome, looking that also involves a lot of math. So that's how I got into the bioinformatics, because that combined my interest in math, computers, and biology. And so that's where a lot of the math majors go for environmental science.
Excellent, thanks.
Yeah.
Any of the other speakers want to add to that?
I can chime in there. So at various parts of my talk, I showed these infographics, right, which are basically life cycle analyses of certain products, like how much water it uses. All of these are basically mathematical algorithms. You're taking the end-to-end life of a compound and walking it through the product phase and its use, and you're putting constraints around it: how much, where, to what extent, what volume, what is its residence time here, where will it be transported, what is the cost of this, what is the waste generated, how much, till in what time frame. These all get sort of like compacted into sort of mathematical constraints that get plugged into some sort of large algorithm where you plug in these values and it computes sort of like both like a techno-economic numbers around a process, as well as a life cycle numbers around this process. These kinds of analysis give a lot of ground truthing to everything that we do. And so those kinds of research also require a lot of math background. And then I told you about, like being able to understand the genome of an organism and its metabolism. Well, really the parts that we understand is the reactions, A getting converted to B, B to C, C to D, and so on. It's a network. A carbon compound comes in, it goes into hundreds of different reactions, which themselves go into hundreds of more reactions, which come together into a node, then bifurcate again into many reactions. Each one of these reactions can be given a rate and a flux, and then that builds what is called a genome scale metabolic model. That's pretty heavy computationally as well. It would be very hard for someone to do the arithmetic or the around it mentally. So that requires computational work, and usually a knowledge in mathematics can be very useful for that. And that kind of metabolic models are built not just for engineering purposes, but also for disease-causing organisms, microbiomes, and other biological processes.
Excellent. Thank you for the very complete answer. Let's go to the next question, and I'll direct this one to Dawn. So what would you say is the biggest environmental impact on people's health? Do you have any suggestions for potential research that I can pursue as I seek to bridge my interests in both health and environmental sciences?
Yeah, that's a great question. So I mean, this is just, you know, broadly speaking, I think that climate change is the biggest threat to our human existence and being able to thrive. Environmentally, it's you know, going to threaten a lot of people's lives and health. And I think it impacts people in California, particularly with who have asthma, you know, from the fires. We now have a fire season, which we didn't have growing up in the Bay Area before. This is just like, you know, what we now experience. Our current lives are just like this now, that you can't go outside during some months. So definitely the impact of climate change is just going to increase, you know, year after year until people start to really put tons of money into solving this problem, or mitigating it or slowing it down. And then I would say for, you know, the combination of environmental science and human health, you know, working in microbiology is one bridge for sure. Microbes are everywhere. They're in your stomach, they're, you know, in the environment. There's people in our lab group that work both on the human microbiome and they also work on plant microbiome. So there's different projects that are all in that. So I think, yeah, you could totally bridge it. You can go in between these areas, you know, for whatever you want to do. It's not, yeah. And also if you work on coding or something like that, those skills translate across, you know, all the fields. So yeah.
Excellent, thank you. All right, the next question.
Could I,
Oh, yeah,
I
go
just
ahead.
want to, I thought Dawn's answer was really great. I just want to build off of that. So, just like a comment about the increase of asthma from the fires. So what they've found out so far is that there's these pollen particles that usually don't cause like a immune response because they're too big, but the fires actually they're so hot that they break up the pollen particles and then they get deeper into your lungs and then cause that asthmatic response. So that's actually like a mechanism that people have figured out that is impacting human health from these fires. And then also people are getting more interested in the impact of the forever chemicals in the environment because they've been finding them in fish and in the waters. And so that's where regulations come in because I think they just passed some new regulatory levels on these forever chemicals because like we're eating all of the fish and the other organisms that live in the water and then they're getting into our bodies. So anyway, those are just some other examples of if you're interested in it. So there's definitely like many links to human health and environmental science.
Yeah, thanks for that addition. Any other follow-ups from the committee? All right, any follow-ups from audience members? Any questions or comments? Okay. Then we've got another question, which is I'll direct to Colby. I think all of you kind of answered this a little bit by giving us a glimpse into your day-to-day life, but the question was, what does an environmental scientist do? Which is very broad, so.
Yeah. So I see science as a really amazing adventure. So, you know, what is an adventure? An adventure is you're going into the unknown where no man or woman has ever been before or thought about before. And it's so exciting, you know, and so that's exactly what a scientist does. A scientist is an adventurer that explores and pushes new frontiers and makes new discoveries of things that could be really important, or could just be academically important, but is making new discoveries that nobody has ever made before. And it's such a special and exciting thing to do. And literally every year I'm making so many new discoveries that I can't imagine doing another type of work where, where you is not based on adventuring and discovery. And so that's, everybody has their different opinions about what a scientist does. For me, it's about being an adventurer and discovering and making new, exciting connections. And going, we're talking about interfaces, like, for me, like I had a problem where like I was studying these VOCs as in the atmosphere. And so everyone was considering me an atmospheric scientist, but I was trying to understand them to understand the biology. And I had a really hard time with the biologist considered me an atmospheric scientist, and the atmospheric scientist would consider me a biologist. And so like I had a hard time like fitting in, but like now I'm getting it kind of figured out. It just took me a while. But yeah.
That's cool that you're like the translator between the two. But I really like your description of scientists, because everything you said applies to me as a particle physicist, and I do a completely different thing than you all. So great description. Does anyone else want to add to that?
Well, I can give a more mundane answer to what a day in the science looks like, depending upon where you are in your career. So at my day mostly is about the nicest part of my day is when I get to sit with the rest of the research group and we are talking about some experiments that has been done in the day before, the weeks before, and we are talking about reminding ourselves of, okay, remember you said that, if you make this edit in this gene, it would have this outcome. Well, that didn't happen. Instead, this other thing happened. What do you think? And what measurements should we make? Those kinds of discussions, those are fun. But then there's other discussions too. There's discussions around how do we run the lab safely. There are safety meetings. We sit and we talk about safety procedures, best practices. We come up with protocols that other people will agree to and follow. We look at things that we haven't done well and try to correct them. Then I go back, and then a big part of my job is to keep a lookout for what kind of government agency is trying to fund what research and how does that connect to mine. I have to do a lot of reading, and I have to talk to a lot of people to make sure that I don't fall out of touch with that. That's sort of my piece of it. I have to make sure that I find the right advocates for my science and that I keep track of their calls for proposals, like where they have said, if this is my topic, this is how much money you can get, make a proposal for a science. And then I write those proposals. This gets passed around a big team of people who contribute their little bits. It goes out to some budget office, a personnel office. They clean it up, make sure that we're following all the rules, and those get submitted. Then we wait. Sometimes we get the money. Most of the time we don't, but it educates us and we move forward and we make more interactions. And in the middle of all of this, nowadays we are very eager to make sure that we are not missing out on a diverse set of opinions and expertise. We are always on the lookout of wanting to work with people from diverse cultural, scientific, and educational backgrounds to make sure we have these big, vibrant teams and that we are doing the right things to support them. All that takes a lot of work too. So your day kind of gets broken down into talking about the research, finding funding for the research, nurturing and being a good steward of the funds that you found, and on and on you go through these phases and these cycles and so on. So that's what my day looks like.
Excellent, thank you. All right, we have a couple of really good questions, and we're running out of time. So if people have to leave, that's fine, but I'd like to have a chance to answer these questions. So let's go through them, but we'll try to keep the answers short. So the first one is to everyone in the committee or the panel. What are the most effective ways to conserve natural resources like water, land, and biodiversity in order to balance economic development and environmental protection? I don't know who wants to start.
I can share my screen for just 1 minute and just show these students a resource. This is a resource built by a colleague of mine who does life cycle analysis, and this is called the JBay biosighting tool. So this might be pertinent to the question that was just asked. Very briefly, not everything works everywhere. Not should everything be tried to be implemented everywhere. What works in California won't work in the Midwest. So a good place to start to answer your questions is to figure out what makes sense where, so that we are optimal in our technology implementation.
Fantastic. Is there a link or something to that you could put in the chat for people to look at?
I will do this.
Thanks.
I
Anyone
could
else?
just add one thing to that, which is it's important that, you know, we become advocates for the environment, because it's going to take more than our individual actions. You know, I feel like politicians kind of put it on the individual, like, well, you got to recycle, you got to, you know, this is a big problem. We have big problems, and we need to advocate for the people who have billions of dollars, or the people that can actually solve these problems, to bring attention to it. So whatever we can do on our social platforms, or, you know, in our individual lives, talking to people about how this is an issue, you need to, you know, this is very serious, and people from my generation care about this. I don't know, I was recently thinking how, you know, we do so much advocacy work as scientists too, to the people we mentor, but I'm like, I feel like almost like talking to Taylor Swift or something would be more effective, you know, if she, sometimes that's how I feel. But anyway, yeah.
Well, maybe you should try.
I did think about that. I'm like, she's, yeah, DMing her.
Yeah, that would be amazing.
Maybe I could jump in there too and just mention that, you know, I think one thing that we could all do is just go outside and connect with nature as much as possible. My uncle Ray Jardine has written a lot of books about how to connect with nature, and you know, anywhere from like going on short walks in parks to really long distance backpacking across the Appalachian Trail, or going kayaking, or just hanging out with friends outside. And I think that the more time that we spend doing that, the more that we love it and we want to protect it and appreciate it. You know, if you see a bear in Yosemite National Park, you know, you'll forever change you, and you'll care about bears in California if you see them. And so I think that there's something that we can all do, and with our time and our money and so forth, is just try to spend more time outside. And I know it's very difficult with a lot of you know, things that are going on in everybody's lives, but I think that's one thing that we could all do more, is try to have a stronger connection with nature.
Excellent. All right, let's move on to the next question, which is, which government policies have been effective in protecting the environment and supporting renewable energy? And how can the policies be improved? What is the impact of suburban sprawl, and how do we make cities more sustainable? These are great questions.
So I already shared that tool. I would say that's a good start. I can take a first stab at that, and then other folks can chime in. So government policies will be a critical piece. There is no doubt about that. This is because off the bat, you know, it's going to be expensive. So subsidies will be required to nurture the adoption of these technologies. Without it, they will be fighting an uphill battle with the current prices of petrochemicals, which are very cheap, right? So the government policies are absolutely, so the low carbon emission policies in California have been very helpful in this matter. A lot of green companies can afford to set up shop in California because of these policies, and they cannot afford to set up shop somewhere else because of the lack of such policies in other places. So yes, there are policies. They're not uniform nationally. They should be, and they should keep an eye not just on the environment, but also on the economy and the needs of the people. Otherwise, you're kind of setting them up as opposing things that politicians can use as an either-or situation, where in reality they are the same thing. Our environment is our environment, right? So they always get set up as an either-or thing. Like, you are doing this green stuff at the expense of my lifestyle. It should not be that. It should be the same thing. And that is possible. And so we need to go towards that. The kind of tools that I shared with you will show you that there is no silver bullet. Depending upon where you are, what grows there, what needs are there, you have to customize your application space. So invoking an example that worked somewhere and saying that exact model will work here will set you up for a losing argument. People will be able to show you immediately that you're wrong. This won't work and that won't work. And that wasn't your point in the first place. So you have to be careful when you debate those points ever. And the third thing I will say is really ask a lot of questions. When someone says recycle plastic, ask them which plastic, where, and how. Educate yourself about those little triangle symbols and really read up about which plastics are and are not compostable. And when they say compostable, do they mean the next 100 years or in the next few months? And what's actually practical, because that will give you the teeth to have some solid conversations where you can make some really good points and come up with solutions that actually work instead of just something on paper, for people to check off and comfort themselves.
Excellent, thank you. Anyone else want to add to that?
So I think the most successful policy that comes to mind is actually the one that stopped refrigerants, changing the refrigerants, because that actually led to closing of the hole in the atmosphere. And so I can't remember what the name of the chemical was, but in the refrigerants, like, they actually showed the mechanism of it destroying the atmosphere. And so this was like highly successful. There is pretty complicated why this actually ended up working, but changing the refrigerants over
Oh, was
time
it
actually
freon?
helped. Huh?
Is it Freon? Freon, the chemical.
Oh,
that-
yes. Okay, Freon, yeah. So that was like, I think that's actually something I think everyone should look up, because that was like kind of it was like something that was agreed on like globally to try to fix. So I think that that was successful. I think one thing also to keep in mind is actually looking at the impact of each of our individual our actions. So one thing that actually contributes a lot to greenhouse gases is just riding on an airplane or driving. And so that has more impact than trying to change like how we eat. So I think having all of the impact from like what California has done to try to set up the infrastructure for electric cars, like, so the infrastructure for some of the stuff has to be there. So there's a lot of articles about like people in the Midwest trying to drive electric cars and then they can't charge them anywhere. And so we have to have the infrastructure and the policies to actually set that up. Some of the things with the like using reusable bags, I think is good, but the overall impact of actually making each of those bags is more than just making the plastic bags. And so there is a trade-off there. So that's something that, more studies have to go into. I think it's good that we don't have as much plastic, but also the energy usage is higher. So, and that's just like a balance of it.
Excellent,
I really want to point out, for everybody, what Lauren just said, that no matter which decision you make, there's going to be a trade-off. There is no solution that just works for everything. So say we eliminate all plastic, poof, gone. There's going to be so much tremendous food waste that it's immediately going to offset any good we ever did, right? So you have to find a solution for that before you go in one direction. So having more of a holistic understanding of what that solution space, where that lives in and where that's applicable, can be very useful. Similar to the example Lauren said about the Freon, there are examples where flame retardants were banished from fabrics. There used to be a point in time where everybody's clothes were full of these flame retardants. You can imagine why. You know, you didn't want to get set on fire, but these flame retardants happen to be carcinogenic. So then by policy, those were taken out. Again, trade-off. You know, somebody tried to do something good without taking into consideration the bigger picture of the implementation. And so we keep going kind of back and forth on that. And that's a very important consideration.
Yeah, very good point. Thank you. All right, I know we are about 10 minutes over schedule, so I think we should wrap it up. But if there are any last comments that the panel members want to make, I would welcome you to do that now. Okay.
I would just say that, you know, if you guys, I don't think you guys need to, you know, worry about your career direction necessarily in high school, but just don't rule things out and just talk to as many people as you can, you know, who are in different backgrounds, because you don't really know where you'll end up, you know, and you don't have to be good at math or coding or whatever at this point in your life to end up in a certain place. You can learn anything you need to know. So yeah, so just I would just say keep an open mind about your future.
Thanks, Dawn.
Yeah, I think to build off of what Dawn said, I think scientists are always teaching themselves and learning new things. I think also like what I think Colby has a very diverse background, and he was talking about being an engineer and scientist. Like you're constantly learning and teaching yourself. And so I think that's very empowering to try to learn how the world actually works. And also kind of like what Andrilla was talking about, interacting with the policymakers. I think, you know, using that information to make a change is really exciting. So
Thanks, Lauren. Anyone else?
Yeah, I would just say that, you know, you got to follow your own instincts and not necessarily what everybody else is doing. And sometimes you have to do the hard thing that, you know, like go to college in a different country or state that you don't have friends or family, and you really do have to kind of just follow your instinct, because you need to kind of choose and figure out your own path. And your path may be very different from other people. And you should use other people as inspiration, and also, you know, have some you know, mentors that you can come to with questions and ask them what do you think about this or that. And so try to find, and I think that's a great aspect of this program, is that you're meeting a lot of mentors that you could potentially stay in touch with as you start to move into college and so forth. So yeah.
Really great point. Thanks, Colby. Andrilla, you want to finish up?
No, I mean, very good points were made. I wish you all the best, and you met like 4 new people today. Don't hesitate from reaching out to any of us with questions in the future. We are all on LinkedIn as well, so please connect and let us know if we can ever help in any regard, even if it's something small.
Thank you. And I want to thank the 4 of you. I learned so much today, and I really appreciated all of your talk. So thank you. That was great. And thanks to all of you who joined today. I hope you also learned a lot. And I think Danny's going to share a slide that we can, there we go. Okay, so our next SAGE is going to be in August. We take the months of June and July off to do our summer camps. And so here's the information on that. It will be about internships, some opportunities for you. And you can follow us if you want to get a reminder when it comes closer to August, and learn some stuff when we post other cool information. You can follow us on various social media. And as Andrilla said, feel free to reach out to us and connect. And that's what we have for today. So thank you all.
Thank you.